Good morning to everybody that is connected to this Zoom. I am Riccardo Dalla Favera. I'm a professor at Columbia University, and I have the honor and the pleasure to chair this symposium showcasing the 3 finalists of the Young Investigator Paola Campese Award, which is sponsored by ISSNAF, the Italian Scientist and Scholar North American Foundation. ISSNAF as a foundation that has a mission to empower, connect, and celebrate the Italian intellectual diaspora in North America and foster a sense of shared identity and culture among its members. It has been founded in 2007 and now counts over 3,000 members, US and Canada. As part of its activity, ISSNAF sponsor a number of awards in all field of knowledge, physical sciences, medical and biological sciences, humanities, and so forth. And today the symposium, as I said, will showcase a finalist of the Paola Campese Award for research in hematologic malignancy. This award was established by Stefania and Vito Campese in 2011 in memory of their young, talented, and generous daughter, Paola. Today we will listen to the presentation of the 3 finalists that have been selected by a selection committee chaired by me and composed by Professor Simona Colla, who is at the University of Texas Mandy Anderson Cancer Center, and Professor Laura Pasqualucci at Columbia University. They selected the 3 finalists, which will be further evaluated based on their presentation by a jury, which is chaired by me and would be composed by Professor Colla again, and by Professor Caligari Scapio, that is the scientific director of the the Association of IRC, Association for Italian for Cancer Research, the major founding body of cancer research in Italy. With this introduction, I will open the symposium to the speaker, and the first speaker will be Dr. Giulia Biancon, who works as a postdoctoral associate at Yale University Medical Center, School of Medicine. The title of her presentation will be Uncovering Pathogenic RNA Mechanisms in Splicing Factor Mutant Hematologic Malignancies. Please, Dr. Biancon, you can go ahead with your seminar.
Thank you so much. I'm gonna share my screen now. Okay. Okay, so yeah, thank you again for the introduction. I was a little bit nervous, but actually now after talking with you all before, I feel better. And as Dr. Dalla Favera introduced, the topic of my research is the pathogenic RNA mechanism in splicing factor mutant hematologic malignancies. And before going further into the details of my project, actually, I prepared a few slides, just 2 slides in Italian. I don't know if my family members were able to connect, but I prepared those slides for them. Ok, so il mio progetto di ricerca si basa su 2 concetti fondamentali. Il primo concetto è quello dello splicing dell'RNA, che è il processo di taglia e cuci che determina la diversità cellulare. Le nostre cellule hanno circa 20.000 frammenti di DNA che vengono codificati dall'RNA per produrre fino a 1 milione di diverse proteine. E quindi com'è che passiamo da 20.000 frammenti di DNA a fino a 1 milione di proteine diverse? Riusciamo a farlo proprio grazie al processo di splicing. E qui ho cercato di spiegarlo in modo abbastanza semplice. Quindi abbiamo una molecola di RNA che contiene informazioni utili che però sono sparse in mezzo a informazioni inutili che vanno rimosse. Quindi quello che fa il processo di splicing è che taglia via queste informazioni inutili, mette insieme, cuce insieme in modo funzionale le informazioni utili per poi avere la produzione finale delle proteine. Quindi abbiamo il Duomo di Caorle, la Via dei Pescatori, che è dove c'è la nostra casa di famiglia, e il Rio Terà, che è il centro di Caorle. Il problema è che se ci sono delle alterazioni a questo meccanismo di splicing, questo può portare allo sviluppo di tumori. In particolare io qui a Yale mi occupo di due tipi di tumori del sangue, di mielodisplasie, che poi nominerò MDS in inglese, e leucemia mieloide acuta, AML in inglese in acronimo. Cosa sono mielodisplasie e leucemia mieloide acuta? Sono delle forme di tumore in cui il midollo osseo, che vedete qua, accumula delle cellule immature che sono alterate e non produce abbastanza cellule del sangue sane. Quindi vuol dire che i pazienti hanno un ridotto numero di globuli bianchi, per cui aumentato rischio di infezioni, un ridotto numero di globuli rossi, quindi i pazienti sono anemici, e un ridotto numero di piastrine. Questo causa eccessivi sanguinamenti spontanei. In realtà la leucemia mieloide acuta è una forma più aggressiva delle mielodisplasie. Circa il 30% di pazienti con mielodisplasie sviluppano poi leucemia mieloide acuta e purtroppo la sopravvivenza è molto bassa per questi pazienti. Quindi vuol dire che nonostante i vari processi di miglioramento delle strategie terapeutiche, dobbiamo ancora capire veramente qual è il meccanismo patogenico alla base. Quindi appunto la domanda è: qual è questo meccanismo che è legato alle alterazioni di splicing che poi porta allo sviluppo tumorale? And now I'm going more in details into my research project, and so this part is going to be in English. And so specifically in our lab, in the Halin lab, we are focused on U2F1 splicing factor mutations in MDS and AML. U2F1 is a splicing factor that, together with U2AF2, forms the U2AF complex that recognizes the 3' splice site here, recruiting then the U2 complex that is necessary for the spliceosome activation. We know in the context of MDS and AML that there are two heterozygous hotspot mutations in the two RNA binding domain of U2F1, and specifically they affect the residue 34 here in orange and the residue 157 here in blue. We know that these mutations, they determine RNA splicing alterations that are ultimately responsible for ineffective hematopoiesis. But how these mutations disrupt the ordered splicing and the biological consequences of these mutations remain to be fully elucidated. So I applied a multi-omic approach in order to investigate the aberrant RNA mechanisms driven by U2F1 mutations. And specifically, we wanted to address three main questions. The first question was how U2F1 mutations alter RNA binding, considering that they are in the RNA binding domains of U2F1. And so we developed and applied the fractionated eclipse-seq. We also investigated the relationship between binding and splicing alterations. And finally, we investigated the biological consequences of these mutations. And to do that, we applied immunofluorescence imaging and single-cell RNA-seq. We used different cellular models for our analysis. We used human erythroleukemia cells, health cells, expressing U2F1 wild type or mutant. But we also used primary cells from MDS and AML patients with or without U2F1 mutations in order to validate and translate our observations also at the patient level. So let's start with the binding analysis. First, we modified an approach that already exists, and it's called the eclipse-seq. Is enhanced cross-linking immunoprecipitation, followed by deep RNA sequencing. The advantage of this technique is that it allows to analyze the RNA binding profiles with a single nucleotide resolution. And how do you do that? Simply you map and count the end of each eclipse sequencing grid. And in this way, you can build the binding profile. But then we introduce the membrane size fractionation right after immunoprecipitation. Basically, you cut the membrane in different fractions according to the molecular weight of the protein of interest. In this way, you can isolate the binding signal of a specific RNA binding proteins within a multi-component complex. So for the first time, we were able to isolate the RNA binding signal of U2F1 here in what we call the light fraction, where we have a sharp peak, you can see here, insisting on the A of the AG dinucleotide, right before the 3' splice site. And we isolated the U2F1 signal from U2F2 signal that you can see here on top on the heavy fraction. You can still see the sharp peak of U2F1, but together with a broader U2F2 signal that is insisting on the polypyrimidine tract. So we now we can really study U2F1 binding in the wild type context. But what about the mutant context? What if we have U2F1 mutations? So we applied FreeClip-seq also in the presence of S34F mutations in orange and K157R mutation in blue. And for the first time, we saw that there were binding changes, in particular with a novel peak in position -3 for S34F mutant and in position +1 for K157R mutant, together with a reduction of the U2F2 signal in comparison with the wild type condition. So we know that U2F1 is a splicing factor. So what about the splicing alterations? We also conducted RNA sequencing and alternative splicing analysis. And as you can see, confirming previous literature, the positions that are affected by binding changes and by splicing changes, they perfectly match, because in the alternative splicing, the exon inclusion or exclusions is determined by the nucleotide sequence in position -3 for the S34F mutant, or in position +1 for the K157R mutant. So we have binding alterations that directly affect the splicing outcome, but of what targets? What are the targets that are directly affected by these alterations? So we conducted a functional enrichment analysis of targets that were affected by both binding and splicing alterations, and we observed an enrichment in terms related to RNA granule biology, but in particular to stress granules. So what are these stress granules? The stress granules are biomolecular condensates in the cytoplasm of eukaryotic cells. And they are constituted by untranslated RNAs here, together with RNA binding proteins, and they condense together under stress to improve the cellular adaptation, to improve the response of the cell to the stress conditions. The increased stress granule formation has been reported in solid cancers as a strategy to enhance the fitness of the tumoral cells. However, it was never explored before in the leukemia context. So considering my binding and splicing data, I decided to perform immunofluorescence imaging in order to visualize the stress granule formations. So I induced my cells, the cell lines, but also primary cells with U2F1 wild type or mutant. Then I stressed my cells because in order to see the formation of stress granules, you need to stress the cells of interest. And then I used, I labeled the cells with G3BP1, that is a known stress granule marker, and with DAPI for the nuclei. And then I acquired Z-stack images. And this is the result. So as you can see here on the right side, the signal of G3BP1 in red is clearly higher, is more intense. And this means that mutant cells, they clearly upregulate the stress granule formation in comparison to wild type cells. And this is statistically significant considering all the replicates and all the fields that we acquired per slide. So this means that the binding alterations that directly affect the splicing outcome of specific RNAs, increasing the availability actually of RNAs that are components of the stress granules. And this leads to an enhanced stress granule formation. That means enhanced stress response of U2F1 mutant cancer cells. Finally, I validated these observations also at the single cell level. So I conducted single cell RNA sequencing of MDS patients with U2F1 wild type or U2F1 mutated. Specifically, I isolated CD34 positive cells, and then I performed 5' single cell RNA-seq, 10x. And first, here we were able to specifically define each cell as either mutant or wild type. So here you can see 3 different populations. We have in orange, dark orange, the mutant cells in the mutant patient. So this means that we were able to find at least 1 mutant read in that cell. Then we have in light orange, the wild type cells in the mutant patient. What does it mean? That we couldn't find any mutated read in the mutant patient, but it may be a dropout, or it may be really a wild type cells. And then the third population is the gray population, and are the wild type cells for the wild type patient. So we conducted the mutational analysis, and now we are able to identify 3 different classes. And then we analyzed the stress granule score. What is the stress granule score? Basically, we considered the average expression of 150 genes that were differentially bound, differentially spliced, and also enriched in stress granules. And then we calculated the score for each of the three populations. And as you can see, the stress granule score in the mutant cells, in the mutant patients, it's significantly higher. So it really means that also at the single cell level, we can connect the U2F1 mutant genotype with an enhanced stress granule formation in the mutant cells. E adesso torno indietro alla domanda in italiano che chiude, diciamo così, il significato di quello che ho visto nel mio studio, è che quindi qual è il meccanismo che è legato a queste alterazioni di splicing porta alla fine allo sviluppo tumorale. Ci sono delle alterazioni dello splicing che modificano la composizione e l'aggregazione di specifiche molecole di RNA. Queste molecole di RNA aumentano la formazione di granuli che potenziano e alla fine avvantaggiano le cellule tumorali nel midollo osseo. E questo apre tutto un nuovo campo nello studio, ma anche nel trattamento di mielodisplasie e leucemia mieloide acuta. Of course, now the future plans are based on these discoveries. So I would really like now to understand the effect of other splicing factor mutations in the stress granule formations in the context of MDS and AML, because this may be a unifying oncogenic mechanism. I will also apply different techniques in order to identify the crucial proteins and RNAs that are involved in the enhanced stress granule formation. I will evaluate changes in other biomolecular condensates. And finally, the idea is trying to target both the spliceosome machinery and the biomolecular condensates that are altered. Throughout all these years here at Yale, I have collaborated with many of my colleagues, but also other PIs here, in order to fight together blood cancers. And specifically, I am involved in the RNA sequencing analysis of acute megakaryoblast leukemia. I also developed the pipeline, and I'm in charge of the DNA sequencing analysis in the context of clonal hematopoiesis of indeterminate potential, mast cell leukemia, multiple myeloma, and IDH1 and 2 mutant MDS and AML. I have collaborated to develop in vivo models to study red blood cell disorders and hematopoietic development. And finally, in the last 2 years, I've been involved in the development of an immunoserological assay in order to study the COVID-19 severity in hematological patients. And with that, I would really like to thank the Halin Lab, in particular, Dr. Stefanie Halene, because she's like the best mentor ever, and she taught me so much. I'm super grateful. And of course, also Dr. Toma Tebaldi, that was my mentor for all the computational part. And I would like to thank my amazing collaborators here at Yale. And of course, I would like to thank my family. And thank you for listening to my presentation. And now I'm happy to take all of your questions.
Thank you very much.
Yes,
thank you very much.
Sorry.
Sure. Simona, you can ask.
Sorry, Dr. Dalla Favera, sorry. I have a comment and then a question.
Sure, go ahead.
Okay. As you know, Giulia, MDS became my field. I was in multiple myeloma, so this section is really for me because multiple myeloma and then my MDS. Yeah. And, okay, so comment. Dr. Stefanie Leningiel is my friend, and this is a comment. Every time she spoke, I was with her in Nashville.
Oh yeah.
Every time she present, she always say, "This is the work of Dr. Giulia Bianconi."
Oh.
She created this project. She did this project. This is her thinking.
Thank you so much.
And this is super important because it means that you are independent. Because when a PI go to a meeting, I'm a PI, I go to a meeting and say, my project is for this because this guy, I publish this because my postdoc, Dr. Len, said this to you. And so I absolutely happy that you are a PI and you will be a good PI. Second question. So this is a comment. Then my question is this. Splicing factor mutation. We still did not understand what they are, correct? So I can tell you something. I came from a telomere dysfunction, and my previous work was multiple myeloma. I came from multiple myeloma. I want to work on telomere biology. Telomere biology, develop MDS. And then I figured out this. I saw that if you have a mouse model of telomere deficiency, this mouse model develop MDS, and that's why I'm a passionate for MDS. And I also saw that was a completely down regulation on the splicing factor. And then I started reading literature, and I saw that the granular formation was already published before, but was a consequence of splicing factor downregulation, particularly SFRS2. When you decrease SFRS2 in the cell, you get a formation on granule in the cell.
But the question, Simona.
The question is, so how you could connect this? Because you are talking about mutation, but the issue is that it was already published in terms of downregulation of splicing factor. So in your system, is the fact that the U2F1 induced downregulation something that induced granule formation? Because if you read the literature, was already absolutely published before.
Okay, Julia?
Yes. Yeah, sure. So the downregulation was of the wild-type isoform, right? They didn't conduct studies about mutations, I guess.
Downregulation of splicing factor.
Okay, yeah, so in my case, what I see is that if you have U2F1 splicing factor mutations, you have aberrant binding and also splicing of the same transcript, because it's something that is happening at the same transcript level. And these transcripts, they are longer, they have a longer 3' UTR, and they are reported in stress granule datasets as components of stress granules. So my hypothesis is that we have alternative splicing, we have an increased availability of isoforms that tend then to form stress granules, and this is the reason why we then see the increased stress granule formation. For example, if there is an alternative splicing of what will be an intrinsically disordered region that affect if that transcript can form stress granules or not. And so this is the effect that I see in the presence of the mutations, that actually they don't down-regulate the expression of U2F1, but it's more a gain of function, actually, of the U2F1.
Or loss of function.
Or loss of, sure, or loss of function if we look at it in the other way. Yes, sure. Exactly. So this is what I see in a U2F1 model, both cell lines and patient samples. And now I want to check on SRSF2 actually, because we need to check if it's something specifically only for U2F1 or also in SRSF2.
Exactly, it's not specific for a U2F1. You need to look at the other splicing factor, because this is a overall general mechanism, because it has been already exactly.
Yeah, and plus would be interesting also maybe in lung cancer, because for example, U2F1 is also frequently mutated in lung cancer. So would be nice also to see in another model if it's
okay, consistent. Sorry for the question. We should
this is great discussion, really good. One just short follow-up to Simona's question. As I understand, there are many mutations in different splicing factor in MDS. Do you have evidence, or you think that this will be a general mechanism for all of them? I think you just started discussing this.
Yeah, because of course I talk about SLSF2 because I've conducted a meta-analysis so far and just some preliminary slides that I've still to finish to acquire the microscope. And actually in the meta-analysis, the stress granule signature, it came out. So I think that there is an involvement, plus SLSF2 mutations, they really affect HNRMPA2B1, that is a stress granule component, and this is pretty relevant, already demonstrated. And in my slides, I still see a change. It's not that clear as for U2F1, but I need to finish the analysis, and then I need also to check patient samples, because at the end, we also need to validate on primary cells the observations.
Okay. Thank you very much, Julia.
Thank you, Julia.
Thank you so much for the question and the discussion.
Okay. And now we move to the second presentation from Anna Maria Gullà, who has recently moved to the Candiolo Cancer Institute in Candiolo. But I think she's presenting work performed at the Dana-Farber Cancer Institute at Harvard Medical School. And the work, the title of her presentation is Dissecting the Path to Immunogenic Cell Death in Multiple Myeloma. Please go ahead, Anna Maria.
Thank you so much for the kind introduction. I'd really like to thank the scientific committee for being selected as a finalist for this prestigious award, first of all. And then I'd like to take this opportunity to show you the work that I have done in the past years at Dana-Farber Cancer Institute in the laboratory of Kenneth Anderson, trying to explore and characterize the role of immunogenic cell death in multiple myeloma. First of all, I wanted to give you a brief introduction of the topic and just explaining why I think this is an important topic in the context of the disease. As you know, multiple myeloma is still an incurable malignancy. So what we know, among other things, is that immune dysfunction characterizes the disease since the precursor stages. And several players are involved in these immunosuppressive roles. As you can see here, several have been characterized. But one I wanted to point today your attention is the interplay and the relationship between cancer cell and antigen-presenting cells. This relationship, which is not still well understood in myeloma, is pivotal, is essential, as we know, for an efficient activation of an adaptive immune response and recognition of the tumor. What we have learned from these other cancer type is that some chemotherapeutic agents are able to stimulate and to activate this interaction and to specifically kill cancer cell in an immunogenic way. This specific type of cell death is called immunogenic cell death, and this is associated with a series of molecular events that includes ER stress, autophagy, viral mimicry state in the cells, and the cell death itself, that are associated with a series of danger signal, the so-called damage-associated molecular patterns that are perceived by the immune system, specifically in this case by dendritic cells and T cells, simulating and leading to an activation of the immune response. What we have after the induction of the SED process is a recognition of cancer cell by dendritic cells, phagocytosis of cancer cells that are dying by these chemotherapeutic drugs, and the presentation of the antigens to this and increase the therapeutic efficacy of the drugs. So what we have done so far was to show that bortezomib, which we know is the mainstay of myeloma, not just kill multiple cells, but it kills them in cells dying upon treatment with bortezomib, exposed several DAMPs, and among them, I just wanted to point your attention to carreticulin, this intracellular protein that is exposed on the cell surface of cancer cell, and specifically drives the phagocytosis of cancer cells by dendritic cells. Cells dying by several cell lines, and we saw in patient cells were indeed engulfed by dendritic cells. And this was subsequently associated with an activation of the T cell response. And we saw how T cells were indeed specifically primed against the cancer cell and able to recognize and kill them. And more importantly, what we found was that we showed that this effect was a long-term effect. And after what was also described in the past in other tumors is that treating with an immunogenic drugs is similar to what happened with the vaccination process. So indeed, what we found was that when we treated mice with tumor, immunocompetent mice in a syngeneic model, we of course obtain in this case a strong anti-tumor effect and a regression of the tumor. But these mice were not just treated, but also they were protected against the subsequent re-challenge of live tumor cells. And when we ex vivo analyzed the response of their T cells, we found how they were specifically able to recognize the multiple myeloma cells. So this is consistent with what we did with the therapeutic success. And what we did was to trying to also prove that the therapeutic success that we have with bortezomib in myeloma relies also on the activation of this immunogenic response. And indeed, we identified this immunogenic cell death signature that when we applied to several data set of multiple myeloma patients, and here I'm showing you just the IFM DFCI clinical study, was clearly divided the patients in several subgroups, and patients with a high level of the signature were indeed also the same that were having the best overall survival over time. The deep analysis of the signature led us also to identify an additional mechanism through which bortezomib is able to increase the immunogenic microenvironment. And in this case, we showed that bortezomib is, as we know, able to induce genomic instability in cancer cell. And this genomic instability is associated with the formation of micronuclei. So this extranuclear DNA is sensed by the CGAS STING pathway. And indeed, this pathway was strongly activated intratumorally in cancer cell. The activation of even in the data set of patients, the ICD signature was strongly correlated with STING. And we found how the bortezomib effect on the T cell activation was STING dependent, as when we knocked out STING within the cells intratumorally, we found that the effect of bortezomib on the T cells was significantly decreased. And this was important because, as we know now, STING is a therapeutically actionable target, and several drugs are now clinically available, and among them also in clinical trial in cell hematological malignancies. So what we did was to try the combination of bortezomib with the STING agonist also in myeloma, and to find out this combination was also significantly effective, and how the combination also was dependent on the activation of the immune system, as it was abrogated in the absence of the immune system, and similarly abrogated when we deleted intratumoral STING, so dependent on the intratumor activation of STING. So the nice part of the work was also to find that all these events were dependent on the activation and the exposure of calreticulin on cancer cell. Because when we try to reproduce the same data in a myeloma system in which we knocked out STING, we found both in vitro and in vivo that this effect were completely abrogated. So as was also found in other tumors. So we asked whether any multiple myeloma related alteration may in some way also affect the calreticulin exposure pathway, thus in this way mediating the resistance to immunogenic cell death. And in order to do so, we try to overlap and to integrate several data. So first of all, we try to understand the interactome of calreticulin in multiple myeloma after the induction of ICT. And we perform our mass spectrometry analysis of calreticulin immunobinding proteins in wild-type cells. And we at the same time try to integrate this list of proteins with also genes that we know are important in myeloma and that are connected and correlated with the survival of the patients. We indeed analyzed a list of differentially expressed genes between patients with long survival, between patients with very low survival, less than 1 year. And what we found was these genes that we didn't know was correlated with the ICD process, but we know was a putative, predicted binding partner of carreticulin, GABARAP. GABARAP was specifically higher in patients with the long survival, at the same time specifically binds to carreticulin upon treatment with bortezomib. And it was interesting to note how in GABARAP knockout cells, we were able to completely subvert carreticulin interactome after treatment with bortezomib. And that also show how carreticulin was not binding anymore to proteins on the surface of the cells, but mainly retained within the ER and the mitochondria. So we checked how the outcome of the correlation of these genes with the outcome of multiple myeloma patients. And we found how low level of the genes were correlated with poor clinical outcome. But at the same time, we needed to know the independent role of GABARAP from the deletion of 17p. As we know, deletion of 17p characterized patients with high-risk multiple myeloma, and GABARAP is specifically located on the deleted arm of this chromosome. And when we excluded the patients of 17p from the analysis, we also found our low level of GABARAP were still correlated with poor outcome, thus showing an dependent risk prediction. So our hypothesis was that patients with low GABARAP, and particularly those with deletion of 17p, may not undergo ICD upon bortezomib treatment. And we tried to test this hypothesis. First of all, we were able to see a stronger correlation between GABARAP level within multiple myeloma cells with the extent of carreticulin exposure after bortezomib treatment. And as you can see here, cancer cell with high GABARAP level were also able to express properly carreticulin, but cell lines like here, the KMS11 with low level of GABARAP, almost absent level, were not able to expose carreticulin. So we focus on this specific cell line, which we knew carried the monoallelic deletion of the gene, and we confirmed that indeed carreticulin was not exposed after bortezomib treatment. And at the same time, phagocytosis of cancer cell did not occur after treatment with the drug, although cells were still dying properly, so were not resistant to cell death itself. And interestingly, when we re-overexpressed GABARAP in these cell lines, we were able to both restore carreticulin and at the same time phagocytosis. So to look at the on-target effect of GABARAP, we deleted these genes in several cell lines. And here I'm showing you just one of them to show how the GABARAP knockout clones were not anymore able to expose carreticulin after treatment with bortezomib. As you can see here, cells were still dying in both cases, but only in wild-type condition we can clearly see carreticulin on the surface. And this effect was on target, as by re-overexpressing GABARAP in the knockout clones, we were able to again see carreticulin going on the surface of the cells. As we know, carreticulin is one of the driver of the phagocytosis. So consistently, we also found an abrogation of the phagocytosis process after treatment with the drug in the GABARAP knockout clones. And this effect was specifically linked to the absence of carreticulin on the surface, as by treating exogenously with recombinant carreticulin, we were able to restore the phagocytosis of the cells. Of course, this was also associated with, at the same time, a decrease of the T cell response after treatment with the drug. So T cells after culturing with cells treated with bortezomib but knockout for GABARAP were not able to recognize and kill anymore the T cells. And this was mirrored by the in vivo efficacy of bortezomib that was completely decreased in GABARAP knockout condition. So overall, our work is trying to show how the clinical success of bortezomib relies on the activation of an anti-myeloma immunity, and this occurs through the induction of immunogenic cell death and activation of the cGAS STING pathway. STING agonists may indeed represent a novel therapeutic opportunity in myeloma, either alone or in combination with bortezomib. And we know that the loss of function of GABARAP, and particularly in patients with deletion of 17p, high-risk patients, blocks calreticulin exposure after bortezomib treatment, thus contributing to ICD resistance and tumor immunovation. Here are all the people that I have to thank that worked with me overall the past years. I want to thank my team, but especially my mentor, Kenneth Anderson, for this precious support, all the funding agency that funded my research, and of course, all of you for your attention. I'm very happy to take questions.
Thank you very much, Anna Maria. Thank you very much. It was very clear. Again, Simona, you want to go ahead with a question? I have a couple myself, but go ahead, Simona.
I go ahead or you should go ahead? So
no, you go ahead, but straight to the question.
Okay, it was a super, a lot of data. Just a super, why did you focus on STINGAS? Why at the beginning? Possibly I miss. There are many pathways.
Yeah, so the pathway I was not specific, came out by the analysis of the SED signature. The SED signature was identified by looking at the transcriptome in vivo in tumor after treatment with bortezomib, both in a wild-type condition on calreticulin knockout tumors. This gene signature was specifically used only in tumors in which we had the ICT. And we didn't know what these genes were. So by the analysis of this signature, we identified all the interferon pathway to be strongly activated.
Yes.
And since we knew that bortezomib was inducing genomic instability, we were trying to see also if this was linked to the cGAS STING pathway and connected.
Absolutely, yes, but there are many pathways associated with interferon response. I know this.
Yeah, we checked the NFKB, and I mean, they were not similarly activated. This was the main one that was activated, and especially the one, and because I didn't show for the sake of presentation, whose deletion abrogated the T-cell activation. So when we deleted STING, we did not observe anymore the T-cell activation, while by doing the genome editing of the other pathway, we still observed the effect downstream on the T-cell activation.
Out of curiosity, did you try IF-204? Did you delete IF-204 another?
No, I didn't.
Okay.
Okay.
This is a good suggestion. Thank you so much.
No, this is
just a curiosity. Okay.
Thank you very much. Thank you very much. Super good presentation.
Anna Maria, a couple of quick questions. In the experiment where you show memory, right, where you reinject the cells that in memory, any idea whether there is a specificity in that recognition?
So this is something we're actually working on to specifically look at the T-cell response and trying to identify the specificity of the response. So what we know now is we don't know if the response is polyantigenic, is specific to an antigen. So this was also our question. Unfortunately, I don't have the answer right now, but we had performed several in vivo study by single-cell RNA sequencing and TCR sequencing that are trying to address this question.
Second question is in the relationship with the 17p deletion. Those are monoallelic deletion, right? What about the second allele, reduction of those? Or
yeah, so yeah, what we found was that even with the monoallelic deletion and the lower level of GABARAP, we still have the same phenotype. And indeed, even in patients without the deletion, but with a lower level of GABARAP, we still observe a decrease of the exposure of calreticulin. This is something we're trying to address by using genomic data. But so far, what we observe is that even because GABARAP is lower in myeloma patients in general as compared to normal plasma cells. So in patients with deletion have, of course, even lower level. So our hypothesis is that even the decrease of GABARAP is still able to induce the phenotype.
Okay, perfect. Thank you very much. Thank you very much. Thank you. And so we'll move to the third presentation while he's loading. Dr. Francesco Maura, who is now the assistant professor at University of Miami in the Sylvester Comprehensive Cancer Center. And the title is Deciphering the Genomic Complexity of Lymphoproliferative Disorders.
Thank you. First of all, thanks for having selected my application as a finalist for the Paola Campese Award. My name is Francesco Maura, and as a quick introduction about myself and what brought me to the United States, I did my med school residency fellowship at the University of Milan, La Statale, and I was incredibly lucky to have the opportunity to spend 1 year at the last year of my residency in Peter Campbell lab at the Wellcome Sangris that I extended for additional 2 years. And during all this staying, I had the opportunity to learn how to handle genomic data, in particular whole genome and whole exome sequencing data. Considering my work on multiple myeloma, I was recruited by Ola Langren and Papa Manuel at MSK to start the myeloma genomic lab together with Langren, and then I followed him in Miami, where I'm now. So my lab basically has 4 main areas of expertise, which is kind of like pretty broad, but one is the mechanism of resistance to immunotherapy. One is defining early events, in particular at the DNA level for myeloma precursor condition, and identifying new marker of high resistance and aggressive disease to identify a model and develop model for precision medicine. And we are also very interested in the impact of chemotherapy on tumor and normal cell and how these affect the long-term toxicity. So over this presentation, I will not focus only on one single paper that my lab published over the last 3-4 years, but basically to the idea that is behind our lab, like the core of our research is to basically replace whole exome sequencing with whole genome sequencing. And most of the research that has been done in multiple myeloma and in lymphomas in the last 15 years has been generated mostly on exome sequencing data or targeted sequencing data, which are great technology for coding mutations and for defining copy numbers, but they miss and they're not able to define 2 important genomic features. One is structural variants and one is mutational signatures. So when we talk about structural variants, we talk about like the other side of the coin for copy number changes. So copy number changes is when a chromosome can acquire a deletion or a gain, but when we look in array or an exome data, these are just independent segments that are lost and gained. Thanks to whole genome sequencing, we can look at the breakpoint and link together these different alterations. And so what it seems to be like different independent copy numbers often are part of a more complex event. This one, for example, is a Hodgkin lymphoma genome we sequence, and you can see there is a double menu with multiple copies, up to 15 copies of JAK2 and PD-1 locus. And there are other copy numbers around that seems to be independent, but thanks to the genome, we demonstrate that this is actually one single event acquired in time. The second concept is mutational signatures that they can be summarized as mutational processes behind the cancer evolution, which mutational processes are active in each cancer type and in each. And I will spend a little bit more time on mutational signature in one of the following slides. So most of our research and publications were focused on multiple myeloma. I think Maria presented quite well what is multiple myeloma in her introduction. So it's a post-terminal center tumor, and it's quite interesting, not just for multiple myeloma and hematology, but in terms of cancer evolution, because it has 2 well-recognized clinical entities that precede multiple myeloma in all patients. One is called smoldering, and one is called MGUS, monoclonal gammopathy of undetermined significance. Now, the difference between these 2 entities is clinical. MGUS have a very low risk of progression, while smoldering have a heterogeneous risk of progression over time into multiple myeloma. And the differentiation is based on criteria that have been established 40 years ago by Bob Kiley at the Mayo Clinic. So with our work and our future work, what we are trying to do is to use the genomic events that myeloma acquire over time to define which are the patients that progress and the one that remain stable. Considering that MGUS, monoclonal protein, can be found in up to 6% of the population above 60 years old, thanks to the new technologies we have, this is something important because it's important to predict who is gonna at risk of progressing and who is just have an indolent and benign entity. So to do that, we work around the concept of whole genome sequencing-based genomic defining events, which means genomic events that define multiple myeloma, and they are not present in precursors that will not progress, both smoldering or MNES. So one is using mutational signatures, in particular a process called ApoBec, and then linking together copy number and structural variants. So mutational signatures is an approach developed at the Sanger Institute several years ago, and basically is, as I mentioned before, is aimed to define which mutational processes are active in each cancer type. So the idea is we classify each single nucleotide variant in our genome in 96 possible classes. So this plot represents the 96 classes. Each bar is a classes that account a certain number of mutations. The 96 classes reflect the 6 possible mutations, C2A, C2G, C2T, C2A, C2C, C2G, and 16 possible nucleotide contexts. So it's 6 per 16 is 96. You can see that this is a whole genome sequencing we published a couple of years ago of a therapy-related AML exposed to platinum. And you can see that the mutation prevalence across the different classes is not random. If it was random, we just would not see any difference between the different classes. Instead, you have these peaks. It looks like a skyline of an American city. And so if you use this analogy, you can think mutation signature analysis using like skylines. So we have on the top New York, London, and Rome on the bottom. And these are like, you can recognize them without any mathematical or EI model. Now, if I mix this picture in a chaotic way, you still are able to recognize New York, London, and Rome. And you can quantify how much of this picture is contributed by Rome, London, and New York. That's exactly how mutational signatures work. So we can deconvolute this skyline in different components. And one of these components, for example, is this profile. You can see these two peaks are exactly the same. And we know from in vitro studies, from in vivo studies, that this one is the mutational signatures caused by platinum. So mutational signatures are not just mathematical profiles, but they can be linked to distinct, extrinsic, or intrinsic mutagenic layer. In this case, is platinum. So ApoBec is the mutational signatures that is found in 80–90% of newly diagnosed multiple myeloma patients. These are the two signatures 2 and 13, and this is what's so-called the matrix profile or the skyline. And it's detected multiple myeloma is the cancer with the hematological cancer with the highest proportion of patients with ApoBec, together with Hodgkin lymphoma, much higher than other B-cell lymphomas, for example. And the important aspect about ApoBec is that patients with high ApoBec tend to have a very poor prognostication. This is was confirmed in a couple of papers from our lab, and this is based on the COMPASS trial. So patients treated with immunomodulatory agents and proteasome inhibitors. Now, if we look in this recent paper currently under review, when we sequence the Manhattan trial, a phase 2 trial where patients were treated with daratumumab, carfilzomib, and alidomide dexamethasone, so immunotherapy with anti-CD38 antibodies, ApoBec still retains its poor prognostication, and patients with high ApoBec have definitely a poorer outcome compared to patients with low ApoBec. The complex event and structural variants I mentioned before, we described those in different papers here, and of course it's like a very dense topic for multiple myeloma, but I'm just focused on chromotripsis, which is the most prevalent and also prognostically relevant marker in multiple myeloma. So chromotripsy is basically when one or more than one chromosome are shattered during one mitosis, and all these pieces are reassembled in a wrong way, generating multiple loss and multiple gain. So this generates the involvement of multiple driver genes. For example, in this case, we have multiple chromosomes involved by the same event that usually occur in one or few mitoses. And you can see that all these genes are involved at the same time. So in terms of evolution, a chromotripsis event involving multiple oncodrivers has definitely a huge impact on the cancer history and evolution. This example is just to show that chromotripsis sometimes also have like hyper gain or gain of function. In this case, we have amplification of these genes up to 9 copies, like focal gain. What is important about chromotripsis is not just the prevalence, which is high, 24%, but is that patients with chromotripsis has a poor outcome, both in terms of survival and both in terms of progression-free survival, always using the COMPASS data. So now we have these genomic defining events using whole genome sequencing. The question now is how we can translate this information to MGAS or to smoldering. Now the problem with MGAS is that we have so few cells that it's impossible to really define, to capture enough nanograms to do whole genome sequencing. So the cellularity is too low. So to solve this problem, we worked with the Sanger and Vito Campese that developed this low input whole genome sequencing pipeline that is able to capture the whole genome sequencing with very high quality, I think identical to the bulk genome, starting from 1,000 or 2,000 cells. So we use that for MGAS, sequence 2,000 to 3,000 cells per patient from 15 patients, combined with other data we have from bulk genome sequencing, and we look at which genomic features predict the progression before these patients reach the high risk. So none of these patients have high-risk disease or high tumor burden. What we look is almost a bimodal distribution, where we have that patients that are stable over time, also across many years, don't have any of these so-called genomic defining events. So there were zero ApoBec, no chromotripsy, and also no other like complex events as structural variants or mutations that we didn't have the time to describe. While patients that progressed, they actually had all of these alterations. In particular, ApoBec was the most prevalent and sensitive marker in terms of progression. We recognize this is a very small study, as only 32 patients were included, plus the multiple myeloma genomes from previous publications. So now we are working and we are leading, my lab is leading this multicentric effort to combine all possible exome and genome sequencing data. We develop mathematical approach to call ApoBec and chromotripsy from exome data in the meantime. And the idea is to develop a new classifier, a new prediction model, where it's not anymore MGAS or smoldering, but is benign precursor myeloma versus myeloma 2B or early myeloma.
5 minutes.
Yeah. So this concept of ApoBec and chromotripsy, as many of the concepts we can find at the genome level, are not just relevant for multiple myeloma, but can be translated and applied also for other hematological malignancies, in particular in the B-cell context, that is where we work. So this paper on the top was published a few months ago in collaboration with the Moffitt Cancer Group, where we look at whole genome sequencing from patients treated with CAR-T anti-CD19, a very effective and FDA-approved treatment that is radically changing our approach to relapsed disease for B-cell lymphoma. So what we found is that ApoBec is associated with very aggressive and unfavorable outcome. Basically, all patients with ApoBec activity progress, as well as presence of chromotripsy was another marker of poor outcome. So this is important because most of the research that has been performed so far in CAR-T was focused on immune microenvironment. But in this paper, together with another paper that was published at the same time from MD Anderson, we show that genomic features are also important and genomic complexity. The one below is a bioarchive paper currently under review, where we characterize the genome sequencing landscape from Hodgkin lymphoma. And you can see that here, chromotripsy, in particular with BFB, breakage fusion bridge events like multiple amplification, is quite recurrent in this tumor and involves multiple important driver genes, as well as ApoBec is actually detectable in 80% of the Hodgkin lymphoma. So I want to end with one of our current interest in the lab is cancer chemotherapy mutagenesis. As I mentioned in one of my first slides, we know that platinum and also temozolomide, the skyline or signatures of temozolomide, are associated with distinct signatures. So you know that when you expose a cell to platinum or temozolomide, those cells will acquire a set of private and unique mutations in this nucleotide context that can be detectable when this clone or cells will expand to secondary malignancy. And this is a very hot topic because in particular in myeloma and lymphoma, the survival has increased so much over the last year. So the worst thing that can happen is that the patient that is cured develop a therapy-related disorder that is usually very hard to cure. So our contribution to this field of chemotherapy mutagenesis is the discovery of melphalan mutational signatures. So my lab identifying multiple publications confirmed by collaborators these signatures. So when you expose a cell to melphalan, you get this skyline of mutations, this profile. And this has been observed on relapsed multiple myeloma. Multiple independent data sets show that. Cell lines, we use cell lines to validate this observation. We found in relapsed B-cell lymphoma, before CAR-T, patients exposed to transplant has this set of mutations. Richter transformation with the Elias Campbell, we found melphalan-like signatures as well in the recent Nature Medicine, and also in post-therapy-related myeloid tumors, which is the last slide of my presentation. This is a work that is currently under review, and what we did is investigate 39 therapy-related myeloids, mostly from platinum and melphalan. And as expected, we found the platinum signatures, the skyline of platinum, in all patients exposed to platinum. But what surprised us is less than half of the patients with exposed to melphalan, like transplanted myeloma that received autologous stem cell transplantation, had the melphalan signature. And so why this happened? These cells, all of them should have it. So after digging into the data, checking for CH in the apheresis, and doing some new mathematical modeling, we demonstrate that this scenario of melphalan negative cases post-transplant is due to the reinfusion of the CH. So instead of having a CH that survives the myeloablative conditioning, the CH is collected during the leukapheresis and reinfused, keeping the melphalan exposure. What was interesting also is that all the patients with the melphalan signatures were all TP53 monoallelic or biallelic loss, suggesting that a CH to survive the myeloablative conditioning needs to have at least one copy of TP53 loss. And this is all our work, and this is the lab with a strong integration with our clinic and our funders. And of course, there are so many people to thank for all these projects and papers that I just summarized the map of Europe and America and Canada, all our collaborators. Really, without them, none of this would have been possible. And thank you all for your attention.
Thank you very much, Francesco. And now the talk is open for questions by the audience, but really by Simona and myself. Simona, you have a question?
No, it was a super good presentation. I really thanks Mauro.
Thank you.
I know Mauro is a super good PI, and I have no question. Actually, I need to learn from him. So thank you very much.
Not sure about that, but thank you.
Okay. So, Francesco, so you refer, of course, to the role of ApoBec that you take collectively. Do you have any idea what in the family is active and whether
So ApoBec is active in many solid cancers, and its activity is kind of a mystery. No one is really able to understand why certain tumors have it and others no. What is
referring to myeloma?
In myeloma, we know that there are two groups of ApoBec. One is what we call hyper-ApoBec, and is driven by MAP. So when you have a translocation of MAP and BAP-B, we know these upregulate ApoBec 3A, the subunit 3A, and this promotes a massive mutagenesis. So if you have a patient with more than 20% mutations due to ApoBec, that patient is MAP. It's really unlikely that it's something else. And all patients with MAP, or most of them, have this pattern. All the other patients, we don't have right now an idea. We have hypotheses, and the hypothesis is that ApoBec is a marker of transformation that the cells require when you start to escape the immune system. And so we are working to validate this hypothesis. Because myeloma develops over decades, it's impossible to collect the samples over time retrospectively. So we're working on mouse model with the lead Bersager and Martacchesi to longitudinally follow these mice, because we found that the Kappa-Mik mouse that develop multiple myeloma, 54% of the mice have a clear ApoBec activity. And so the idea is to model this activity and to understand when this activity starts and if there are immunopopulation associated with that. So that's a work that I hope to be submitted in the next 4 weeks.
So myeloma is a post-germinal center malignancy, right? So do you see AID signatures?
Yes, so we see AID signatures.
And we distinguish them from other ApoBec family members?
Oh, yeah. It's AID signatures is like, I don't have the plot here, but you should imagine ticks in pink and in green. Here, this one is actually non-AID. This one is the germinal center signatures, which is probably caused by polyeta active during the lysosome fast replication of the cells when they expand. While the AID somatic hypermutation, the one that Professor Paola kind of pioneer in this characterization in lymphomas, you cannot find using genome-wide approaches because it's cluster only on IgH or in certain genes. So what we do to characterize AID activity is to run a cluster mutation signature analysis on the region where we expect AID to be. Also, don't expect to see if there is new loci. And you can see clearly that myeloma has a strong AID component. But what is interesting and in line with our knowledge is that all the mutations are clonal and all the mutations are early, reflecting the absence of AID in plasma cells or ongoing AID mutagenesis in myeloma.
Okay,
I
thanks.
have a further question.
Sure.
Because we are talking about AID, and so I came from multiple myeloma. I studied multiple myeloma until I gave up. I like something else, okay? And I got this mouse model of ID3 mouse model, in which I was trying to put the mutation in the germinal center. Zero. So can you let me know, do you think that multiple myeloma is really germinal center disease?
I'm 100% sure about it.
Why we cannot get a mouse model if we put the promoter of ID, AICDA? I spent 5 years of my life on doing that.
So
And I got myeloma only if I put Mike.
Okay.
So the idea, I guess, is it's always difficult to. So we in one of our paper, which we bit complicated to present and compress in a few minutes, but we developed a mathematical model that show myeloma took 30-40 years to develop since the first acquisition of the drivers, and so it's very hard to compress this timeline into a mouse model. I think the advantage of the decapamic mouse model is that it takes time, 12-18 months to get myeloma.
You see, no, my question is the germinal center disease?
But the germinal center is just the initiation. So it's like a phase when the myeloma acquires certain drivers. Like we know hyperdiploid gains are acquired in the germinal center. We know cyclin E1 is acquired in the germinal center. And we know that after that, the cell escapes germinal center. These are unpublished data, but hopefully by next year we will submit this paper. And so the idea is that germinal center is what we call early phase, and then all the rest of the phase is outside the germinal center. It's like bone marrow development. So we actually think that the B memory cell phase of myeloma is quite long, but is also very early in time, almost undetectable for us in human.
Okay.
Thank you very much. Thank you super much. Thank you.
So
Thank you.
now we are ending the symposium. And so I thank all the speakers, all three, great job. And of course, I thank the sponsor, the Campese family. And now the jury will meet in the next days and identify a winner. Will be difficult. All three were good, but so we'll see what happens. And thank you very much again. I wonder whether the administration has any other comment. Enrica, you're muted.
Thank you, Riccardo, for chairing the symposium. Thank you to Simona and the jury, and I want to wish all the best to the finalists. And I hope to see you all on November 7th at the embassy in Washington D.C. For the ISSNAF annual meeting, where the winner will be announced. So all the best to everyone.
Final comment to the guy, because I'm in the middle from Dr. Dalfalever and this guy. Go ahead, guy. You're super good. Go ahead.
Okay.
Thank you.
Thank you.
Go ahead with science. Go ahead with science. It's super tough. Go ahead.
So persevere.
Perceive. Yes.
Okay, Enrica.
So all the best to everyone, and thank you for your participation.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you so much.
Goodbye.
Bye.
Bye.
Bye.